Before we jump into the titillating subject of gluconeogenesis, what does the name gluconeogenesis even mean? Now, breaking it down, it simply means we're going to make new glucose. Gluconeogenesis, which is going to be shown here in this reaction, is basically glycolysis just in reverse. Gluconeogenesis is primarily found in the liver, so the liver over here, because that's where the body is going to be low on sugar, and the liver can use gluconeogenesis to pump out more usable glucose for the body. Now, the pathway begins with pyruvate. So we've got pyruvate down here, and then it ends with our glucose molecule up here, and it's going to use most of the common enzymes. However, remember, from glycolysis, there were three enzymes, so I start them here, that are going to be irreversible. Three irreversible steps of glycolysis, the arrows that have a single head. However, we need to bypass that. So to bypass those three irreversible steps of glycolysis, we need to introduce four new enzymes. Because gluconeogenesis has four irreversible enzymes, we need to have a couple pathways in our first step down here, and then we need a couple enzymes up here to bypass these irreversible steps. So gluconeogenesis, we're only going to throw at you a few new enzymes. Before we look at these four enzymes, I've got a question for you. Do you remember what cofactor is needed for carboxylase enzymes? I had you repeat this one with me a few times, so hopefully you didn't forget. Carboxylase enzymes typically require biotin as a cofactor. Now, why am I even talking about this? Well, I'm probably going to be introducing a carboxylase. So let's bring it in. We've got our pyruvate carboxylase. This is the first key enzyme in gluconeogenesis, our pyruvate carboxylase. This is going to convert our pyruvate to oxaloacetate. This step occurs in the mitochondria. It requires biotin and ATP, and it's activated when there is a high concentration of acetyl-CoA. Since this means you don't need to be producing more acetyl-CoA and ATP, and it would be better off storing energy as glucose in glycogen. Next, PEP carboxykinase is going to convert oxaloacetate into our PEP molecule, or phosphoenolpyruvate. So this occurs in the cytosol. This uses GTP as a source of the phosphate. Fructose 1,6-bisphosphatase is the third irreversible enzyme, and this is going to convert our fructose 1,6-bisphosphate into our fructose 6-phosphate up here. And then lastly, we've got our last enzyme, that's our glucose 6-phosphatase enzyme, and this removes the phosphate from glucose 6-phosphate to form our free molecule of glucose, which is then free to be released into the bloodstream and be used within the body. This last step occurs mainly in the hepatic endoplasmic reticulum, and it does not occur in muscles, since remember, muscles lack this enzyme. Glucose 6-phosphatase is also the enzyme that's mutated in von Gierke's disease, which we'll be talking about pretty soon. So in summary, gluconeogenesis occurs primarily in the liver here. It's going to create glucose from glycolysis, fatty acid, or TCA cycle intermediates. It should become as no surprise that a deficiency in one of these four main enzymes here is going to result in hypoglycemia or a low blood sugar level. How else can we make glucose molecules? Here we see our TCA cycle, which we already touched on. Odd-chain fatty acids can also serve as glucose sources since they yield propionyl-CoA. And this can enter the TCA cycle as succinyl-CoA. Even-chain fatty acids, which we'll see here, however, cannot be a glucose source. Now, why is this? I just said odd-chain fatty acids can be a glucose source, while even-chain fatty acids cannot. Now, this is because they are broken down into acetyl-CoA, which disappears in the form of carbon dioxide by the time it reaches oxaloacetate. Remember, oxaloacetate was used in gluconeogenesis as one of our starting points for converting oxaloacetate to glucose. So, by the time that these even-chain fatty acids get broken down, we're going to lose both carbons in the form of CO2, so therefore it can't be used to make glucose. Now let's talk about the HMP shunt. The hexose monophosphate shunt, aka the pentose monophosphate shunt, is shown here. Its purpose is to synthesize NADPHs and ribose sugars. NADPHs and ribose sugars. Now, ribose sugars are 5-carbon sugars. So, when glucose 6-phosphate is going to be abundant, we can shunt it to make these products. Now, in the irreversible oxidative phase, so here, we see one NADPH is going to be produced by the first enzyme, glucose 6-phosphate dehydrogenase, and then another is going to be produced later on. Do you remember what NADPH is used for within the body? The NADPH can be used in reductive and anabolic reactions, such as glutathione reduction in red blood cells and cholesterol synthesis in the liver, which is why the pathway is most active in lactating mammary glands, the liver, adrenal cortex, and red blood cells or erythrocytes. Now, if the cell already has plenty of NADPH, this pathway will shut down since NADPH inhibits the rate-limiting step G6PD. Without NADPH, cells are much less capable of handling oxidative stress, but we'll talk about that more in a minute. If cells need a lot of NADPH, they can make this pathway into a cycle by converting fructose 6-phosphate into glucose 6-phosphate. This pathway consists of an oxidative, irreversible phase, followed by a non-oxidative, reversible phase, both of which take place in the cytoplasm. Now, the irreversible steps create the NADPHs. So here we see another NADPH step being created. Now, there's also a reversible non-oxidative phase where the HMP shunt also produces a ribose 5-phosphate in the process. So this process uses transketalases, which can then be used for a nucleic acid synthesis. Remember, our ribose sugars are used in nucleic acids. Alright, the transketalases require vitamin B1 and are often used to test for B1 deficiency. Now, notice how the end products here of the HMP shunt can get fed directly back into the glycolysis pathway. Our end products are fructose 6-phosphate and G3P. Now, both of those are intermediates of the glycolysis pathway. This way, the body can use the HMP shunt to make NADPHs or ribose derivatives without actually creating much waste. Let's test your knowledge. A 2-year-old girl is born with a glucose 6-phosphate dehydrogenase deficiency, or a G6PD deficiency. This interferes with which step of the HMP shunt? Well, we just covered it, so hopefully you're thinking this is going to disrupt the irreversible oxidative phase, or the first reaction of the HMP shunt. This first reaction converts glucose 6-phosphate into CO2, 2 NADPHs, and a 6-phosphogluconolactone molecule. As promised, now let's talk about the respiratory burst. Now, besides some anabolic reactions, NADPH molecules are also used in the respiratory burst, or the oxidative burst if you want to call it, in which reactive oxygen intermediates are produced by immune cells, normally in the body, like neutrophils and monocytes. Now, these kill invading bacteria or fungi. Now, the first step is the activation of a membrane-bound NADPH oxidase. This is going to convert free oxygen, which we breathe, and combine it with NADPH to form an oxygen radical. So this is going to produce the superoxide radical. Superoxide dismutase then converts this superoxide to hydrogen peroxide, which can then either be chlorinated by myeloperoxidase to form hypochlorite, which that you may know as bleach, or it can be detoxified by a catalase enzyme to make water. Since this hypochlorite kills bacteria, it's going to be a useful pathway for neutrophils and monocytes, but hydrogen peroxide is also harmful to the host cell So the enzyme that we just talked about, the glutathione peroxidase, is going to be used to neutralize it. What happens if a patient is missing this first enzyme, this NADPH oxidase? Well, these patients develop chronic granulomatous disease since they can't produce the superoxide radical or the hypochlorite to kill bacteria. They can still use the hydrogen peroxide that's going to be produced by the invading organism to make the hypochlorite, but they can't synthesize it on their own. So they're still going to have a high risk of infection by catalase-positive organisms. Alright, because these species will have their own catalase to convert the hydrogen peroxide into water. So a catalase-positive organism will be able to convert its own hydrogen peroxide into water, so the body can't make hypochlorite to get rid of it. I'll go over part of this pathway on the next slide. If a patient comes in with recurrent positive catalase organisms, what should you be thinking? You need to start thinking about a respiratory burst deficiency.